Today I am taking up the part 3 of digestion and absorption, that is physiology of digestion or the process of digestion. So what is the definition of digestion? I have given here. Digestion means breakdown of complex organic molecules into simple, soluble and absorbable products due to the chemical and mechanical action in the digestive system. That means the complex organic molecules are broken down into simple substances that can be absorbed into the blood and used in various metabolic activities. So in the digestive system, food is subjected to two types of actions. Mechanical action, that is mastication in the mouth or in the buccal cavity with the help of the teeth, churning movements in the stomach, and also peristaltic movements in the intestine. So all these processes pulverize the food into small pieces or bits. Chemical action, it is brought out by the action of the various types of enzymes that are secreted in the digestive glands. Now let us see how the process of digestion occurs. Digestion always begins in the buccal cavity. So in the buccal cavity, I have told you there are three pairs of salivary glands. The salivary glands secrete slightly acidic saliva. Saliva contains one enzyme, that is salivary amylase or ptyalin. It acts upon the starch and converts starch into maltose, a disaccharide sugar. Here we have to remember one point. All the digestive enzymes are known as hydrolases. Hydrolases means they use water in their actions. So I have drawn here water. In other reactions also water is involved, but it is not represented. Then about 30% of starch present in the food is converted into maltose in the buccal cavity. The rest is digested in the intestine. Then there is another enzyme called lysozyme in the saliva. Lysozyme is a powerful bacterial or bactericide. That means it destroys bacteria. So the bacteria present in the food is destroyed by the lysozyme present in the saliva. Then the food passes into the stomach in the form of bolus. In the stomach, with the arrival of the food, the hormone gastrin is released. The hormone gastrin acts upon the gastric glands, and as a result, the gastric glands release digestive juices and also mucus. The mucus, along with bicarbonates, forms a layer, or it lines the cavity of the stomach and protects the stomach wall from the action of the HCl and other enzymes. Then the oxygen cells, I have told you, they secrete a strong or highly concentrated hydrochloric acid. The hydrochloric acid serves a number of functions. First, it destroys the bacteria. Next, it provides alkaline medium to the food. Acidic medium to the food. Otherwise, the food cannot be digested in the stomach. Next, it activates pepsinogen into pepsin. Pepsinogen is a protein digesting enzyme. It is secreted in inactive state. So inactive pepsinogen is converted into active pepsin due to the action of hydrochloric acid. Then the activated pepsin acts upon the proteins and converts them into proteases and peptones. Then in babies, the main food is milk. Milk contains a protein known as casein. But that casein has to be digested. For that, in the stomach, one enzyme known as prorenin is secreted. Prorenin is an inactive enzyme. It has to be activated into renin by the action of the hydrochloric acid or HCl. So, on activation, renin acts upon casein, the milk protein, in the presence of calcium ions and converts it into calcium paracasinate and proteases. So, in this way, casein present in the milk is converted into a semi-solid calcium paracasinate and proteins. Then, I have told you the oxytic cells also secrete Casil's intrinsic factor or just intrinsic factor. It is very important because it helps in the absorption of vitamin B12 from the food. Otherwise, there will be vitamin B12 deficiency leading to some types of anemia. In this way, in the stomach, of course, some lipase is also secreted. Its action is not that important. Mainly, proteins are partially digested into proteases and peptones. Vitamin B12 is absorbed. Then, casein is converted into calcium paracasinate and proteases. That means only partial digestion of food takes place and that food is in acidic medium. So, at the end of digestion in the stomach, we get chyme. Chyme is nothing but partially digested food in acidic nature. This chyme is slowly released into the duodenum. When the chyme enters the duodenum wall is activated and it releases 4 hormones. They are cholecystokinin, gastric inhibiting peptide, secretin, and enterocrinin. Cholecystokinin acts upon the gallbladder and contracts it. As a result, bile juice is released into the duodenum. Gastric inhibitory peptide, it acts upon the stomach and prevents further secretion of gastric juice. Secretin acts upon the pancreas and helps in the secretion of watery fluid rich in bicarbonate. Enterocrinin acts upon the pancreas and helps in the secretion of pancreatic enzymes. And also intestinal enzymes. So in this way, with the arrival of the chyme into the duodenum, bile juice is released into the duodenum. Pancreatic juice is also released into the duodenum, and the intestinal glands, namely the Brunner's glands and crypts of Lieberkuhn, also release their secretions. Remember, in the stomach, food is digested in the acidic medium. In the small intestine, food digestion occurs only in alkaline medium. So, first of all, the acidic medium has to be removed and alkalinity has to be produced to the food. For this, the bile juice, pancreatic juice, the secretions of small intestine, all of them have bicarbonate. The bicarbonate removes acidity and provides alkalinity. Otherwise, the food cannot be digested in the small intestine. Now, coming to the bile juice, its pH is 8.4, that is, it is alkaline. Though we call the liver as the largest digestive gland, it secretes no digestive enzymes. Its role in digestion is also very limited, but it has a number of other vital functions, which we are not concerned here. The bile salts present in the bile juice, they emulsify fats. They break larger globules into smaller globules so that the enzyme lipase can act effectively on fat molecules. Then bile salts also help in the absorption of digested lipids and also fat-soluble vitamins. Then the bicarbonate present in the bile juice provides alkalinity to the food. This is the role of bile juice as far as digestion is concerned. Then coming to the pancreatic juice. Its pH is about 8.4, just like bile juice. It has a number of enzymes. They act upon all the three types of food materials, that is proteins, fats and carbohydrates. First, trypsinogen. Trypsinogen is also an inactive enzyme. It has to be activated into trypsin by another enzyme known as enterokinase. Enterokinase is secreted in the small intestine. Enteron means intestine. So enterokinase, which is secreted in the small intestine, activates trypsinogen into trypsin. Then there is another enzyme in the pancreatic juice known as chymotrypsinogen. It is also secreted in inactive state. Chymotrypsinogen is also an inactive enzyme. It is to be activated into chymotrypsin by trypsin. So first trypsinogen is converted into trypsin, then this trypsin converts inactive chymotrypsinogen into chymotrypsin. Then both trypsin and chymotrypsin acts upon still undigested proteins, proteases and peptones and converts them into dipeptides and amino acids. In this way, proteins, still undigested proteins, proteases and peptones are converted into dipeptides and amino acids. Then the lipase. Lipase acts upon the emulsified fats and converts them into diglycerides and monoglycerides. Then starch is there in the food. It is the form of polysaccharides. Amylase, the enzyme amylase acts upon the starch and converts into disaccharides. Disaccharides are three types: maltose, sucrose and lactose. Each one is made of two hexose molecules or monosaccharides. Disaccharides are made of two monosaccharides. Then the DNA, RNA are also digested by the action of the pancreatic juice. So the enzymes acting upon DNA and RNA, or simply the nucleic acids, are known as DNase, RNase. DNase and RNase converts DNA and RNA into nucleotides. Nucleotides are the building blocks of nucleic acids. So in this way, food is digested due to the action of pancreatic enzymes. Now let us see the role of succus entericus or intestinal juice on digestion. The pH of succus entericus is 7.5 to 8. That is, it is also alkaline in nature. Then, as I have told you, enterokinase. It activates trypsinogen into trypsin. Then, there are dipeptides, dipeptidases. Dipeptidases in the succus entericus. The dipeptidases act upon dipeptides, these dipeptides. And the dipeptides are converted into amino acids. In this way, the proteins are ultimately converted into amino acids. Amino acids are the end products of protein digestion. Then, I have told you three disaccharides are formed: maltose, sucrose and lactose. Maltase acts upon maltose and converts it into two molecules of glucose. Sucrase acts upon sucrose and converts sucrose into one molecule of glucose and one molecule of fructose. Lactase acts upon lactose. Lactose is the milk sugar. And it converts lactose into one molecule of glucose and one molecule of galactose. In this way, the disaccharides are converted into monosaccharides. Disaccharides are converted into monosaccharides. So, the glucose, fructose and galactose are the end products of carbohydrate digestion. Then, I told you nucleotides are formed due to the action of DNAs and RNAs. These nucleotides are converted into nucleosides by the action of nucleosidases. Nucleosidases. Then these nucleosides are converted into sugars and bases, nitrogen bases, by the action of nucleosidases. So in this way, nucleotidases and nucleosidases convert the nucleosides into sugars and bases. Then, di and monoglycerides are there, formed by the action of the lipase of the pancreas. Then here, the lipase of the intestines digests the diglycerides and monoglycerides into fatty acids and glycerol. So fatty acids and glycerol are the end products of lipid digestion. So at the end of digestion, what happens? From carbohydrates, we get glucose, fructose and galactose. From proteins, amino acids. And from lipids, fatty acids and glycerol. These are the end products of digestion. Remember, water needs no digestion. Similarly, vitamins also need no digestion. Then, these digested food products have to be absorbed into the blood. Otherwise, the very purpose of digestion is wasted. Now, let us see how the digested food is absorbed into the blood. So, I have told you the end products of digestion are monosaccharides, amino acids, glycerides, and fatty acids. Absorption of digested food mainly takes place in the jejunum and the ileum. Then, absorption of the digested food takes place in 4 ways. Now let us see what are those 4 ways. Passive absorption. Passive absorption requires no energy expenditure and the substances move against the concentration gradient. So from higher concentration to lower concentration, the materials move. So through passive absorption, amino acids, small amounts of glucose, some electrolytes like chlorides, and water and chlorides are absorbed. So through passive absorption, amino acids, some glucose, some electrolytes are absorbed. Then what about water? Water moves across osmotic gradient. Water movement also requires no energy expenditure and it moves according to osmotic gradient. You know what is osmotic gradient. Then second one is active transport. It involves expenditure of energy in the form of ATP. It is a costly process. In active transport, substances move against concentration gradient. That means they move from lower concentration to higher concentration. That's why it involves expenditure. The substances that are actively transported are amino acids, monosaccharides like glucose, electrolytes like sodium. So these are actively absorbed into the blood. Then in the book, the substances absorbed in different parts of the digestive system are given. See, in the mouth, certain drugs are absorbed. In the stomach, water, simple sugars, and alcohol are absorbed. In the small intestine, it is the main site of absorption. Almost all the digested food is absorbed into the blood in the small intestine. Large intestine, water, some minerals, and drugs. They are absorbed in the large intestine. Then the third type of absorption, it is known as facilitated absorption. That means the movement of substances are assisted by some other substance. Some other substance gives assistance. So it is known as facilitated diffusion. Here, the carrier ion involved is sodium ion. Sodium ion participates in facilitated diffusion. Remember, facilitated diffusion involves no expenditure of energy. No expenditure of energy. Then what are the substances transported through facilitated diffusion? Fructose and some amino acids. So these two substances are transported through facilitated diffusion. Then the fourth type, fatty acids and glycerol. These are the end products of lipid digestion. Being larger in size, these molecules cannot diffuse into the blood. Not only that, they are insoluble in water. Because they are insoluble in water, the fatty acids and glycerol cannot be absorbed directly into the blood. So their absorption is in the indirect way. It is like this. Being insoluble in water, they cannot be absorbed directly into the blood. These are first converted into micelles, which move into the intestinal mucosal cells. So inner layer of the intestine is called mucosa. So these fatty acids and glycerol are first converted into micelles, and micelles are transported into the mucosal cells. In the mucosal cells, the micelles are converted into chylomicrons. So when the micelles are coated by protein substance, the micelles are converted into chylomicrons. These chylomicrons are released into the lacteals present in the villi. So in the lacteals, lymph is present. When the lymph is loaded with digested lipids, the lymph appears milky in color or whitish in color. So lymph loaded with digested lipids is known as chyle. So in this way, digested food is absorbed into the blood through four means. That is passive absorption, active transport, facilitated diffusion, and transport of fatty acids and glycerol. So in this way, the digested food is absorbed into the blood, and undigested food is sent into the rectum, colon and rectum. Food is stored there for some time, undigested food is stored there for some time, and that is excreted in the form of feces through anus. So in this way, digestion of food takes place in the alimentary canal of human beings. That is about digestion. Thank you.